welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Biotechnology and its applications. And we are in part 1 of the series. All right, so let's begin. Okay, so question number 1. BTCotton variety that was developed by the introduction of toxin gene of Bacillus thuringiensis, BT, that is resistant to, this appear in NEET 2020, pretty recent. A, insect pests. B, fungal diseases. C, plant nematodes. D, insect predators. Okay, so it was developed so that it is resistant to A, insect pests. All right, answer is A. Let's go to the discussion. Okay, so BT toxin. Okay, BT comes from this word Bacillus thuringiensis. All right, contains a toxin for insects obtained from bacillus. So the story is such that this bacteria was found to contain a gene called cry gene. Okay, and yeah, if you can remember it, it was a crying moment for the insects because they found out that this cry gene encodes for a protein which is toxic to insects. Okay, it is toxic to insects. So those protein crystals that were formed were toxic to these insects. All right, so now once the toxin is ingested by these insects, okay, they were actually the protein crystals that were the toxins were actually in the inactive form. Initially they were found to be in the inactive form, but when they were ingested by the insects, they got activated and then eventually killed the insect. Okay, now how that happened is that they were activated by the alkaline gut. Okay, so the gut of the insect is alkaline in nature and this activated the toxin. So from protoxin became into activated toxin. Okay, and then it bound to the surface of the midgut epithelial cells. It creates pores and cause swelling and lysis, eventually killing the insect. Okay, so this was a very revolutionary way of protecting the crops from insects. Usually farmers don't want their crops to be destroyed by insects because then of course it affects their yield, and then the whole agricultural industry will be affected. So because of that, they discovered this. The scientists, the researchers discovered Bacillus thuringiensis, which encoded for a particular toxic protein, and then that was eventually used in crops. Okay, so to answer your question, yeah, so BTCotton is used. So there are some genes, cry genes, that encode in BTCotton. Then there is also for maize. Okay, there are cry genes that are encoded for maize. So those are BT maize or BT corn. Okay, so they are effective towards those particular insects that affect that yield of the crop. Okay, all right, so but to answer your question, yes, it was against insect pests. Okay, let's move on. Question number 2. What triggers activation of protoxin to active toxin of Bacillus thuringiensis in bollworm? So when NEET 2019, A, acidic pH of stomach, B, body temperature, C, moist surface of midgut, D, alkaline pH of gut. So we already discussed in the previous question. It's alkaline pH of gut. Okay, answer is D. So again, I've already explained that the cry gene that encoded for that particular protein crystal, which was toxic in nature, right? And it is inactive. Therefore, it does not affect the bacteria itself because it's in inactive form. It gets activated only in the alkaline nature. So insects have an alkaline gut. Okay, it has an alkaline gut, and so that converts the protoxin, which is in the inactive form, to toxin. Okay, Yeah, so that pretty much summarizes how this protein crystals work and how BT cotton and BT corn were created. And yeah. Yeah, what triggers the activation of protoxin to active toxins, alkaline pH of gut. Okay, so that answers our question. We can move on. Question number 3. Which of the following is true for golden rice? Okay, and this appeared in NEET 2019. A. It has yellow grains because of a gene introduced from a primitive variety of rice. B. It is vitamin A enriched with a gene from daffodil. C. It is pest resistant with the gene from Bacillus thuringiensis. D. It is drought tolerant, developed using agrobacterium vector. Okay, so what is true for golden rice? It is B. Option B is the right answer. Let's go. So golden rice, as you can see from the term itself, it's golden. Okay, it's yellowish in color. Okay, and it's a rich source of vitamin A. Actually, it's a rich source of beta carotene. Okay, beta carotene. Okay, which then gets converted to retinol in humans, and that is vitamin A. So, especially in those countries where there is a deficiency, a nutrition deficiency, this golden rice is very useful, okay, to cover that malnourishment. Yeah, now it contains two genes. First one is Psi, that's phytoene synthase from daffodil, okay, and there is CRT1, phytoene desaturase from soil bacterium Ermina uridovora. Okay, so it is a combination of these two genes which forms the golden rice. So the yellowish color that you see is from the daffodil. All right, so if you can remember the names of these two genes, it will be helpful if they were asked in the exam. But of course, surprisingly, this information is not present in NCRT. You didn't see it anywhere in NCRT. So I assume it's important since it was asked in NEET, and I would suggest you remember this. Okay. All right, but to answer your question, yes, it is vitamin A enriched. Yes, we know that. And with a gene from daffodil. Okay, clear. Let's move on. All right, so question number 4. In RNAi, the genes are silenced using, this appeared in RISA NEET 2019, A. dsRNA, B. ssRNA, C. ssRNA, D. dsRNA. So ds is double stranded and ss is single stranded, all right? So in RNAi, that's RNA interference, the genes are silenced using A, double-stranded RNA. Okay, so the answer is A. Let's go to the discussion. So RNAi is RNA interference. So you are, as the term itself suggests, you are interfering with the RNA. Okay, so we know that mRNA is translated into proteins. Yeah. So what if you want to interfere with this RNA? Then it will not translate into that protein, right? So you can get rid of something that you don't want. Okay, so basically that's the idea. So RNAi genes are silenced using double-stranded RNA. Okay. Now, usually RNA silencing was found to be a very natural phenomenon that occurred in eukaryotic organism. Okay, it was a form of cellular defense to protect itself. Okay, it destroyed mRNA that it didn't want. Okay, so that was a form of defense for the eukaryotic organism. Okay, it was found that it naturally occurs. Okay, now what happens is this double-stranded RNA is cleaved in those single siRNA, single interfering RNA. Okay, so if you have this double-stranded RNA, it is cut. Okay, so you have these now small portions of RNA, double-stranded RNA. Okay, and this is done using the dicer enzyme. Okay, now this unwinds and attaches to RISC. Okay, let me use a different color for this. Okay, let's say this is RISC. RISC is RNA induced silencing complex. Okay, another enzyme. So it will attach over here. I'm using the same color, sorry for that. It will attach over here, okay, and bring it to the mRNA. So for example, if this is your target mRNA that you want to get rid of, that you want to silence, okay, it will attach over here. And then what happens once it attaches itself over here, this RISC siRNA, it attaches over here, it will silence this mRNA, so that will not translate for that particular protein. Okay, so that way you have disengaged or disenabled that mRNA to translate for that protein by attaching another mRNA to it, which you really don't need. Okay. Yeah, so it just continues what I've just said. So the siRNA RISC binds to target mRNA and prevents translation. So basically this is the idea between for this RNA interference, and like I said, it naturally occurs in eukaryotic organisms, but now they have used that same principle in using it for plants, even nematodes. For example, tobacco plants are affected by nematodes. Okay, and so because to avoid that destruction of the roots of tobacco plants, they have then used this RNA interference in the nematodes to stop that translation from happening. So that is one of the applications that it was used in, and we'll come to that now in the future as we move on with the questions. Okay, so basically I'm just like giving you an example of where it's actually used. So in tobacco plants, yeah, it is used to prevent infection from the nematode Melodigene incognita, which attacks the roots of the plant. Okay, so yeah, this is it. This is what basically RNA interference is. Okay, let's move on. Question number 5. Which part of the tobacco plant is infected by Melodigene incognita? So you just discussed this, appeared in 2016. A stem, B root, C flower, and D leaves. So it is B root. Okay, so tobacco plant roots are affected by this nematode. Okay, roots of the tobacco plant are infected by Melodigene incognita. All right, let's move on. So it's pretty direct. Question number 6. Golden rice is a genetically modified crop plant where the incorporated gene is meant for biosynthesis of, and this appeared in 2015, A. omega 3, B. vitamin A, C. vitamin B, D. vitamin C. Okay, so golden rice is meant for the biosynthesis of vitamin A. Okay, answer is B. We already discussed the two genes that are involved, right? So they eventually form beta carotene, and then beta carotene is the pro-vitamin A, which gets converted to vitamin A in humans. And yeah, so it's genetically modified crop, golden rice. Okay, that's the precursor for the formation of vitamin A. Yeah, right, let's move on then. All right, so question number 7. In BTCotton, the BT toxin present in plant tissue as protoxins converted into active toxin due to, this appeared in 2015 cancelled exam. A. Action of gut microorganism. B. Presence of conversion factors in insect gut. C. Alkaline pH of the insect gut. D. Acidic pH of the insect gut. Okay, so it is alkaline pH of the insect gut. Option C is the right answer. We already discussed this, so this is just a repeat of questions. It seems like an important topic which has appeared over and over again. BT toxin and BTCotton is a protoxin which is activated in the insect gut. Insect gut is alkaline, which facilitates the conversion. Okay, so this I've already explained it. We'll move on. Question number 8. crops engineered for glyphosate are resistant or tolerant to, and this appeared in 2015. It's a cancelled exam again. A, insects, B, herbicides, C, fungi, D, bacteria. So it is B, herbicide. Okay, so the answer is B. So glyphosate is actually widely used as a herbicide. Okay, so it is used in plants to control the growth of weed. Now the weeds tend to compete with the plants for nutrients. So to eradicate the weeds, glyphosate is used, which is a herbicide. Now uh, it in turn causes plant diseases too. Okay, and hence the crops have been genetically engineered to protect itself from glyphosate. Okay. Now, glyphosate, obviously, yes, it helps in crop management by getting rid of weeds. But on the other hand, it also causes lots of diseases. So now to counter that, they have genetically engineered in such a way that they protect itself from glyphosate in such a way that they are not affected. All right. So, but this information is not there in NCRT. So I would suggest you remember this. All right, let's move on. I think I've answered the question though. Yep. Let's move on. Question number 9. Which of the following BT crops is being grown in India by the farmers? This appeared in NEET 2013. A, brinjal, B, soybean, C, maize, and D, cotton. Yes, it's BTCotton that we know is being grown in India by the farmers. So the answer is D. Yep, BTCotton is grown in India. All right, let's move on. Question number 10. RNA interference involves, and this appeared in Karnataka NEET 2013, A, synthesis of cDNA and RNA using reverse transcriptase, B, silencing of specific mRNA due to complementary RNA, C, interference of RNA and synthesis of DNA, D, synthesis of mRNA from DNA. Just discussed RNA interference. So silencing of specific mRNA due to complementary RNA. All right, so the answer is B. So yeah, this is basically a repeat of what I've just discussed, how RNA silencing works. Okay, so you can go back and watch that part of the video again. Just to brief you, yes, it is a natural phenomenon. Okay, and the double-stranded RNA is cleaved into single interfering RNA, and then that is unwound, and RISC, another enzyme, RNA induced silencing complex, then attaches itself to the mRNA, and thus disengaging or disabling the mRNA to translate into the protein. All right. This is basically a summary of what I have just explained earlier, but yeah, it's the same thing. So, but to answer your question, yeah, it's B, option B. Let's move on. Question number 11. Consumption of which one of the following foods can prevent the kind of blindness associated with vitamin A deficiency? Sorry for my fumbling. A, flower sour tomato, B, canola, C, golden rice, and D, BT brinjal. So vitamin A deficiency is definitely C, golden rice. Answer is C. Yes, so we know golden rice was invented to produce beta carotene and counter the vitamin A deficiency, and administration of vitamin A prevents night blindness in humans. All right, so yeah, to answer your question, prevent the kind of blindness that's actually night blindness associated with the vitamin A deficiency, the answer is C. All right, let's move on. Question number 12. Tobacco plants resistant to a nematode have been developed by the introduction of DNA that produces in the host cells. This appeared in means 2012. Both sense and antisense RNA, A, particular hormone, C, an antecedent, D, a toxic protein. Okay, so it is A, both sense and antisense RNA. Answer is A. So this they are referring to RNA silencing or RNA interference. Okay, So DNA introduced via the vector agrobacterium produce dsRNA, okay, which then silence the target mRNA and hence prevent protein formation. So basically what happens is in the plant, let's say this is your tobacco plant, okay, the single stranded the double stranded siRNA are formed here, okay, all right, with the help of the dicer enzyme. All right, now let's say you have the nematode, you know, the roundworms, right? They come and try to feed on this plant, okay. So now these siRNA will enter here, and the RISC will also be present there, okay. Now they will then bind to the mRNA over here, mRNA over here, and then it will destroy the nematode, okay. So now this mRNA will not get translated to a protein, thus killing the nematode, okay. So basically this is how it works. Now these plants, these tobacco plants, have been genetically engineered in such a way that when the nematode tries to feed on these plants, it will kill the nematode. Why? Because the mRNA is the RNA is being silenced in the nematode and eventually killing it, okay. So that way these plants are protected against the infection of these nematodes over here, okay. So basically this is how it works. This is the principle behind it. Erase this. Now, well, obviously the double-stranded RNA produces sense and antisense RNA. Okay, so the complementary RNA binds to the mRNA in the nematode and eventually killing it, because obviously this mRNA will become deactivated, disabled, and then there'll be no protein formation. Okay, so it kills the nematode at the end. To answer your question, yeah, it was pretty simple, right? Oh, sorry. Let's go back to the question. Yeah, tobacco plants resistant to nematode have been developed by the introduction of DNA that produces in the host cell. I love how they frame the question. So they have been developed by the introduction of DNA in the host cell. Basically, this is how it works. Okay, so they have genetically engineered the tobacco plant in such a way that it produces the sense and antisense RNA. Right, the DNA is modified in such a way that they produce the sense and antisense RNA, and then they eventually obviously affect the nematode, which is our result, which is what we want. So both sense and antisense RNA are formed here. All right, clear. Good question. All right, so question number 13. The process of RNA interference has been used in the development of plants resistant to, this appeared in 2011, A, nematodes, B, fungi, C, viruses, and D, insects. So it is nematodes. We already discussed this so many times. So the answer is A over here. Yeah, so basically this is just a repeat of the previous discussion. So I'm not going to go into details. So it is resistant to nematodes. All right, let's move on. Question 14. Bacillus thuringiensis forms protein crystals which contain insecticidal protein. The protein disappeared in Mainz 2011. A binds with epithelial cells of midgut of the insect pest, ultimately killing it. B is encoded by several genes, including the gene cry. C is activated by acid pH of the foregut of the insect pest. D does not kill the carrier bacterium, which itself resistant to this toxin. It forms protein crystals which contains insecticidal protein. This protein, now what does this protein do? It binds with the epithelial cells of the midgut of the insect pest, ultimately killing it. Answer is A. This is the appropriate answer for this. Is what the protein actually does. Okay. Again, I've already discussed this. so I'm not going to go into detail. So far we have studied RNA interference and BT, the action of the protein crystals in BT. So these two topics are important to understand the concept. All right. So far we've come across these two principles or applications. Sorry. And yeah, I'm not going to repeat this again. You can go back and watch that part again. All right. Let's move on. Question number 15. Silencing of mRNA has been used in producing transgenic plants resistant to, this appeared in 2011, A. Bollworms, B. Nematodes, C. White rust, D. Bacterial blights. Okay, so silencing of mRNA has been used to in producing transgenic plants resistant to nematodes. Answer is B, because it affects the roots of the plant. All right. Question number 16. The GM brinjal in India has been developed for, this appeared in 2010, A. Insect resistance, B. Enhancing shelf life, C. Enhancing mineral content, D. Drought resistance. So GM brinjal in India has been developed for insect resistance. Answer is A. Again, it's the same principle as BT cotton. You have BT brinjal, which is developed as an insect resistant crop. All right. Let's move on. Question number 17. Some of the characteristics of BT cotton are, this appeared in 2010, A. Long fiber resistant to aphids, B. Medium yield, long fiber resistant to beetle pests, C. High yield and production of toxic protein crystals which kill dipteran pests, D. High yield and resistant to bollworms. So some of the characteristics of BT cotton are, of course, they produce high yield and they are resistant to bollworms. Okay, so resistant to that. Answer is D. Alright, so BT cotton, obviously, yeah. So they inject into the toxin gets injected into the bollworm in case of BT cotton. A bollworm is an insect which affects the cotton plants. So now, if the bollworm tries to infect the or try to eat into the BT cotton plant, what happens is it will kill this insect. Okay, because of that toxin that's present in BT cotton. Okay, and then obviously the cry genes are involved in this. There are particular cry genes. I think for cotton it's cry1 AC and cry2 AB for cotton, and for maize it's cry1 AB. Okay, for maize. So there are particular genes that will that encode for that toxin, that particular toxin in that plant, which affects that insect, which affects this plant. Okay, so for example, bollworm, cry1 AC and cry2 AB produce a toxin which will affect bollworm. Okay, and it has been incorporated in cotton. Similarly, cry1 AB will produce a toxin that infects another insect which affects infects, sorry, maize plants. Okay, so that will kill the insect over there. So it behaves like an insecticide. Okay. To answer your question, yes, resistant to bollworms. Question number 18. An improved variety of transgenic basmati rice, this appeared in 2010. A. Does not require chemical fertilizer and growth hormones. B. Gives high yield and is rich in vitamin A. C. Is completely resistant to all insect pests and diseases of paddy. D. Gives high yield but has no characteristic aroma. Of course, it gives high yield and is rich in vitamin E. That's the improved variety of transgenic basmati rice, which we have studied as golden rice. Okay, so the answer is B. Okay, so it's rich in beta carotene. Alright, let's move on. What is true about BT toxin? So we are in 2009. A, BT protein exists as active toxin in the bacillus. The activated toxin enters the ovaries of the pest to sterilize and thus prevent its multiplication. C, the concerned bacillus has antitoxin. D, the inactive protoxin gets converted into active form in the insect gut, which is the right answer. Okay, which is true about BT toxin. We have already studied this, so it's the same principle here. All right, let's move on. Question 20. Transgenic plants are the ones, this appeared in 2009, A, generated by introducing foreign DNA into a cell and regenerating a plant from that cell. B, produced after protoplast fusion in artificial medium. C, grown in artificial medium after hybridization in the field. D, produced by somatic embryo in artificial medium. Okay, so transgenic plants are the ones that are generated by introducing a foreign DNA into a cell and regenerating a plant from that cell. Okay, answer is A. The transgenic plants, from the term itself says trans. Okay, that means it has genes or contains genes that are unrelated to the plant. Okay, and have been incorporated into its genome. All right. For example, golden rice. Okay, you have a normal basmati rice. Now we have incorporated a genes or a pair of genes which produce vitamin A and gives it the yellow color also. I'm sorry. Right, so they are not part of this basmati rice genome, but we have incorporated into it so that we can produce another new form of plant. Okay, a crop. So like for example, BTCotton, same principle here, right? So now BTCotton is not a normal cotton anymore, right? It's a transgenic plant because now we have incorporated this BT gene, the cry gene, into it. Okay, which is producing that toxic protein, and that eventually affects the insects that attack it. Okay, so these are unrelated to the plant and have been incorporated into its genome. See, that's what it is. Okay, so to answer your question, yeah, it is definitely B, C, and D don't fit into the description. All right, this process is done with the help of rDNA. We already discussed the concept of rDNA previously, so you can go watch my videos on biotechnology principles and processes. The rDNA is incorporated into the cell, which then grows into the transgenic plant. All right. Okay, so we've come to the end of this video lecture, and this went by pretty fast, I guess. And I hope you've enjoyed today's video lecture, and I hope we've learned something new today. And please don't forget to like, share, and subscribe to NEET Biology. And I'm available on Facebook, Twitter, Instagram, so you can follow me there for more updates. And I'll see you in the next video lecture. All right. So bye.